Up until now, we have looked at both the structure of eukaryotic cells and how those cells produce and manipulate energy. All of these processes are controlled by enzymes, like kinases, and held together by structural proteins, like collagen and microfilaments. Where do these proteins come from, and what controls their production? The code for these proteins lies in the DNA, a long polynucleotide molecule that is found in the nucleus of our cells. A single DNA molecule can be over 2 centimeters long and contain the code for hundreds of genes. We've seen how DNA is tightly packed into chromatin, and then chromosomes by histone proteins. All the DNA in a single cell is called its genome. These molecules are a code that is passed to the next generation. They are the heritability factor, and all life on Earth uses DNA to transmit its genetic code. This here is a chromosome. Chromosomes are tightly packed molecules of DNA. In fact, each chromosome is made of a very long, single DNA molecule. During most of the cell's life, the DNA is unwound to some degree and looks like a bowl of spaghetti. However, just before the cell replicates, it is all coiled up into this package. Chromosomes have 2 sections, referred to as Q and P, and unique repeating sequences at the ends called telomeres. The Q and P sections are separated by what are called a centromere. Centromeres are regions of a chromosome where special kinetochore proteins bind the DNA. Each chromosome only has 1 centromere. Before cell replication, all the DNA is copied, and we end up with 2 of every chromosome. We call these sister chromatids. They are held together by cohesins, which are proteins that sit along the length of the chromosomes. Additionally, the centromere is what helps them stay together for proper sorting when the cell finally splits. Replicating cells go through a life cycle. This cycle happens at some point to most cells of the body, with a few exceptions. The cycle can be broken down into 2 main steps: interphase and mitotic phase. The mitotic phase is the process of cell division, and interphase is what the cell does between replications. Interphase consists of 3 segments. They are G1, S for synthesis, and G2. G1 and G2 mean gap 1 and gap 2. During the gap phases, the cell is busy doing metabolic things like glycolysis and the citric acid cycle. The S phase, or synthesis phase, is where DNA is replicated in preparation for the mitotic phase. The G1, G2, and mitotic phases all have checkpoints that regulate when the cell can proceed. These checkpoints are molecular signals that make sure the cell is ready for each step. The G1 step is the most important, and the cells that do not get the signal to pass the G1 checkpoint often stop replicating. They have defaulted to the G0 phase, which means they are not replicating but maintaining their metabolic functions. Most of the cells in the adult human body are in the G0 phase. There are many other checkpoints throughout the cell cycle, like the ones that stop cells with DNA damage from completing the S phase. However, if a cell passes these checkpoints, it can enter the mitotic phase, which is where the cell actively divides in a process called mitosis. Mitosis consists of 5 phases, referred to as prophase, prometaphase, metaphase, anaphase, and telophase. Let's take a look at these phases and what is happening inside the cell at each phase. Here we are in the G2 phase. We have just cleared the checkpoint to start mitosis. The main players in mitosis are the centrosomes, which contain 2 centrioles, and the chromosomes inside the nucleus. Remember that centrosomes are control centers for the regulation of microtubule formation. We are now going to get to see what this can do. We start mitosis in prophase, where the DNA is finally condensed into chromosomes. The DNA was replicated in S phase, so we already have 2 copies of each chromosome bound together at the centromere. The nucleolus has disappeared because the DNA is condensed. Outside the nucleus, the 2 centrioles have started assembling microtubules, which are growing outward from each and pushing them apart. These growing spider-like shapes are called asters. These will ultimately grow into the mitotic spindle. As mitosis progresses, we move into prometaphase. Here, the nucleus has begun to break apart. and the centromeres of each chromosome pair are covered in kinetochore proteins. The microtubules are continuing to grow out of the centrioles. Some of them attach to the chromosomes by binding to the kinetochores at the centromeres, and others help lengthen the cell by pushing against their counterparts. Once all the centromeres are bound by microtubules, and the centrioles have been pushed to opposite sides of the cell, we are in metaphase. This is characterized by the appearance of the metaphase plate. The metaphase plate is an imaginary region where all the chromosomes have lined up in the middle of the cell. The important part of this is that each sister chromatid is bound to microtubules from opposite sides of the cell. They are still held together by cohesins, but they are being pulled in opposite directions. Finally, the cohesins are cleaved, and the microtubules pull the sister chromatids apart and away from each other to opposite ends of the cell. This is called anaphase, the separation and movement of sister chromatids to opposite sides of the dividing cell. Finally, the cell enters telophase, where the two sets of chromosomes are at each end of the cell, and a nucleus begins to form around them. The nucleolus reforms as well. Each half of the cell is now a genetically identical copy of the other. This leads directly to cytokinesis, where a cleavage furrow appears and ultimately pinches the cell into two daughter cells, each containing identical sets of chromosomes, now housed within their own nuclei. Humans have two pairs of 23 chromosomes in each of our somatic cells. Somatic cells refers to non-reproductive cells. Basically, any cell in your body that isn't a sperm or an egg cell is a somatic cell. Sperm and egg cells are called gametes. Gametes are haploid cells, which means that they only have half the chromosomes of somatic cells. In humans, that number is 23. During sexual replication, two gametes come together to form one somatic cell. In fact, chromosome number is one barrier to reproduction between animal species. A lion and a polar bear cannot have babies because a polar bear has 74 chromosomes and a lion only has 38. Chromosomes also hold clues to our evolutionary past. Most other ape species have 48, but humans have 46. Scientists have discovered that human chromosome number 2 is actually a fusion of two chromosomes from an ape-like ancestor. Chromosomes 12 and 13 from modern apes likely fused in an early lineage that led to human beings. Scientists have noticed that human chromosome 2 has one main centromere, but also has the remnant of a second centromere, suggesting it was at one time two chromosomes. Additionally, most chromosomes only have telomere regions on their ends, but human chromosome 2 has them in the middle as well. Chromosomes contain evidence that we share a common ancestor with all life on the planet. However, they also show us evolution's power to shape the massive diversity of life we see around us.